In this lecture, we treat biology as a layered system, much like modern software stack. From atoms to organisms, we will map biological layers to concepts you already understand from computing. So let's start our biology stack. Uh, as we So this slide actually gives you the mental map. As we just discussed, biology, like software, is layered. Each layer constrains the ones above it. The analogies are not perfect here, but they are here to orient your intuition. Some of these things we kind of discussed in our first lecture. So these are a little bit more elaborated. Let's start with atomic orbitals. At the lowest level, chemistry is governed by atomic orbitals. Chemical bonding only happens when orbitals overlap in compatible ways. For our drug design, these rules decide whether binding is even possible. Okay, so let's talk about bonds that hold molecules together. Here you can see covalent bonds. They are, strong and stable, and then whereas non-covalent forces, they are weaker and irreversible. So together, these bonds allow molecules to bind, unbind, and change shape. Let's start the analogy, that is logic gates. Um, think of orbitals like logic rules. If the rules are not satisfied, there is no interaction, right? So the logic gates is the same idea. And then let's talk about information flow. Uh, our DNA is about 3 billion base pairs per copy. When a gene needs to turn on, that stretch gets transcribed into RNA that you can think of as a temporary working copy. And then, this RNA that gets translated by ribosomes into protein. The protein does the actual work. This is what, you keep hearing, the central dogma. If you are designing RNA drugs, you are intervening this flow at RNA step. When you are designing small molecule drugs, you are usually targeting the protein at the end. If you are a software person, this should feel familiar. There is a permanent storage, a working copy, and then the execution. The analogy is not perfect, but it is useful for reasoning about biology. But there is one thing, the key difference is, unlike in computers, in biology, so there is no separate code from execution. The program and the hardware are just one and the same. Let's briefly discuss about ribosome. Um, I, as we discussed in our first lecture, the ribosome is nature's just-in-time compiler. It reads mRNA 3 letters at a time. Each triplet codes for one amino acid. There are 64 possible triplets, but only 20 amino acids, so there is redundancy. Some triplets are stop signals, so they terminate translation. We will discuss this much more when we are going to discuss mRNA design. So you just need to have a basic idea right now. A single cell is, in some sense, a self-sufficient, self-maintaining system. It has everything it needs to store information, produce energy, and controls its own processes. The nucleus, it protects genetic material. The cytoplasm is the execution environment where most of the work happens, and the cell membrane works like a firewall, regulating what comes in and out. When you are designing a drug, getting it across the membrane is often the hardest part. These small lipophilic molecules can diffuse through. Large charged molecules need transporters or some other delivery systems. That's why drug discovery is such a big field. Now let's briefly discuss organelles as microservices. Inside the container, there are specialized subsystems, what you call organelles, each handling a part of the workload. You can see some examples here, like mitochondria for power generation. You can think of membrane proteins, they act like API endpoints. Um, let's talk about logistics. Inside every cell, thousands of processes run in parallel. The endoplasmic reticulum and Golgi apparatus, these organelles handle protein packaging and delivery, like a continuous integration system that builds and deploys updates. Meanwhile, the lysosome and proteasome run cleanup cycle. A typical protein lasts from minutes to some hours and days and even months actually before degradation. This turnover matters for drugs. If you inhibit a protein, the cell usually compensates and just make more proteins. That's one reason why drugs need continuous dosing. Since we are discussing that point, we can talk about newer approaches like PROTACs. They hijack proteasome to degrade specific proteins faster. Just a different mechanism. Instead of blocking function, you are accelerating removal. Now let's briefly discuss power. ATP acts like a power source. When ATP is converted to ADP, free energy is released. That energy is then coupled to drive reactions that would otherwise won't happen. So this is how cells, kind of pay for work, like synthesis, transport, movement. So now let's talk about networking layer. So at this scale, cells coordinate in groups, tissues, to form, to perform specialized functions. Same idea, right? Local specialization and global behavior. When you give someone a drug orally, it goes through the gut and into the bloodstream, distributed to all tissues. The liver tries to metabolize it. The kidneys try to clear it. If it hits the target tissue, but also off-target tissue, and that's very important. That's why drugs have side effects. Now let's discuss organisms. An organism integrates many subsystems, immune system, endocrine, metabolic systems, into a decentralized self-healing network. So one very important thing we need to understand, we have what, some 30 to 40 trillion cells, and there is no single central controller. When tissue gets damaged, local cells detect it, release some signals, and then maybe immune cells migrate in. Clotting can happen, then repair starts. So it is emergent from local rules. So this is why biology is hard to model end to end. You can simulate a single protein, maybe perfectly, but that protein is in a cell, which is in a tissue, which is in an organism with immune responses, metabolism, and feedback loops all interacting. Your model predicts binding perfectly, but the drug still fails. Why? Because of all this complexity. Now let's briefly discuss about versions, like CI CD systems. Life evolves through variation and selection, nature's own continuous integration and deployment system. Each generation tests small changes. Successful ones persist. Across time, these updates accumulate and expanding capability instead actually. Why does this matter? Because when you are designing new proteins or antibodies, you start from already evolved scaffolds. You make targeted mutations to existing structures. Okay, now let's see what is your mission. So now that you can see life as a layered architecture, you can start reasoning about it like an engineer. You already get layered systems, APIs, data flow, distributed computing, biology. You just saw, right? Same concepts, just different implementation. When you are building models for drug discovery, you need to know which layer you are working at and what are the constraints in that layer. Different layers need different modeling approaches and different data types. So in this course, we are going to discuss layer by layer what you need to know to build models for each modality. Let's discuss a summary. Biology works as a layered system, just like software. Information flows from DNA to RNA to protein. Your engineering instincts map directly onto these layers, but models must respect the mechanisms at that layer you are working on and the physics and chemistry underneath it. Next, we will step through biology across 18 orders of magnitude and focus on what matters for modeling at each scale. Thank you.